Hey everyone, welcome to the Explainer. Today we're taking an architectural tour of Kingdom Animalia. Our main goal is to figure out exactly how to classify animals based on their shared characteristics and their distinct body plans. Think of this like looking at the ultimate biological blueprints. We're going to explore the exact specifications that separate something as simple as a sponge from a complex mollusk, and really everything in between.
Here's our roadmap for today. We'll start with a welcome to Kingdom Animalia, check out the blueprints of animal body plans, look closely at sponges and cnidarians, and then worms and mollusks. We'll wrap it all up with a quick stop and think knowledge check and a concept recap. Let's get to it.
Section 1. Welcome to Kingdom Animalia. Okay, so before we look at the really complex structures, we've got to ask a basic question. What actual bricks and mortar make up an animal, right? Like what separates us and all the creatures we share this planet with from, say, plants or fungi? Well, at their absolute core, first and foremost, animals are multicellular, eukaryotic heterotrophs. That last word there, heterotroph, is super important. It basically means we've got to get our nutrients from other organisms. We can't just make our own food like plants do. And speaking of plants, unlike their cells which have rigid walls, animal cells totally lack cell walls. This is a massive architectural difference. Because we don't have those rigid walls, animal cells have the freedom to organize into complex tissues. This specific structural freedom is literally what allows animals to adapt, survive, and thrive in incredibly diverse habitats with totally different means of physical support. Plus, most animals reproduce sexually, starting as a single zygote, and most actually have the incredible ability to move independently at some stage in their life cycle. Now, when we categorize these creatures, we usually split them into two massive groups, which you can see defined here. An invertebrate is simply an animal lacking a backbone, whereas a vertebrate is an animal with a backbone. Think of us, we're vertebrates. But the really crucial thing to remember for our tour today is that the phyla we're going to be looking at, they all fall under the invertebrate category. Believe it or not, the vast majority of animal architecture on Earth is actually built without a backbone. Moving right along, Section 2, Blueprints, specifically animal body plans. You know, just like a building needs a solid foundational layout, animals have structural organizations that dictate how they interact with their environment. At the very basic level of classification, true animals can be largely divided into three groups based on their body symmetry. First up, we have asymmetry. This literally means there's no true symmetry at all. It's seen in modern clades like the parzoa, which includes sponges. Next, we have radial symmetry. This is where the body is symmetrical around a central axis, kind of like the spokes on a bicycle wheel. You'll see this in the clade cnidaria, which includes jellyfish. And finally, we have bilateral symmetry, which is found in the largest of the clades, the Bilitaria. This simply means the left and right sides are mirror images of each other. And it's not random, by the way. All of these symmetry types are perfectly suited to the specific evolutionary and survival needs of these animals.
Okay, so for animals that have that bilateral symmetry we just talked about, another major architectural feature comes into play, and it's called cephalization. This is the concentration of sense organs and nervous tissue at the anterior end of an animal's body. Basically, in plain English, it's the evolution of a distinct head. And once an animal has a head, we can actually start mapping its body with coordinates. The head end is anterior, the tail end is posterior, the backside is dorsal, and the belly side is ventral. Cephalization was an absolutely massive evolutionary leap because it allowed animals to essentially meet their environment head-on, sensing danger or food way before the rest of their body even caught up.
Beyond just symmetry and heads, we've got to look deeper inside the blueprint at body cavities. During embryonic development, animal cells go through stages. They form a hollow ball called a blastula, and then they fold inward to form what's called a gastrula. This folding creates specific tissue layers, the outer ectoderm, the inner endoderm, and the middle mesoderm. And this whole process brilliantly illustrates how we categorize complex animals by their cavities. A coelom is a true fluid-filled body cavity that is completely lined by that middle mesoderm tissue. A pseudocolum, on the other hand, is a false cavity. It definitely exists, but it isn't fully lined by the mesoderm. These cavities are super important because they act as protective cushions for internal organs and give the animal systems a flexible space to actually operate.
All right, Section 3, Sponges and Cnidarians.
Let's check out some of the earliest and honestly most fascinating built structures in the animal kingdom. Now, looking at this, you might be tempted to think it's some kind of colorful underwater plant. But nope, it is unequivocally a living animal. This is a sponge belonging to the phylum Porifera. Despite just sitting there looking totally stationary, it takes in food, digests it, grows, and reproduces. Let's break down exactly how this beautifully simple animal actually functions.
The word porifera literally translates to pore bearer, and honestly, that tells you almost everything you need to know about its design. A sponge is covered in epithelial-like cells for protection, but here's the crazy part. Inside, it lacks any true tissues whatsoever. Instead, it relies on just 2 layers of independent cells that are separated by a jelly-like substance. So, how on earth does it eat without a mouth or a stomach? Well, it uses specialized color cells equipped with whip-like flagella. As these flagella whip back and forth, they literally draw water right into the body of the sponge through its thousands of pores. The sponge then filters nutrients from this water, and the leftover water and waste materials are forcefully pushed out the top through a large mouth-like opening called the osculum. It's basically an incredibly efficient living water pump.
Now, let's step the complexity up just a notch and look at the cnidarians, which include your jellyfish and sea anemones. What's absolutely wild here is that most cnidarians have this unique adaptation that is rarely seen in animals of more recent origin. They actually exist in 2 totally different body forms during their life cycle.
Let's contrast these 2 forms, because they're pretty different. On one side, we have the medusa. This is your classic jellyfish shape. It's an umbrella-shaped body with tentacles that hang straight down. The mouth of a medusa is located on the ventral or bottom surface, right smack in the middle of those hanging tentacles. On the flip side, we have the polyp form. The polyp has a tube-shaped body that's designed to stay anchored to a surface, and its mouth actually points upwards, completely surrounded by a ring of tentacles. Both of these forms come armed with stinging cells, but their architectural orientation is completely flipped.
Section 4. Worms and mollusks.
All right, so leaving the radial symmetry of the cnidarians behind, we're now entering the world of bilateral symmetry and much more complex internal cavities. Phylum Mollusca, which includes things like snails and squids, and phylum Annelida, which includes your segmented worms, are some of the most familiar groups of what we call lophotrochozoan protostomes. I know, it's a huge word. They're grouped together because they share a common ancestry, specifically tied to aquatic species that have a very distinct larval stage called a trochophore larva. But the biggest takeaway here is that these animals possess a true coelom. Having this fully lined, fluid-filled cavity allows for incredibly flexible and diverse body plans. And honestly, this structural flexibility is exactly what has led to their massive biological success and abundance all over the globe.
Section 5. Stop and think.
Let's hit pause on our architectural tour for a quick second to check our understanding. Here is a super common misconception, especially when you're first studying biology. Think back to what we've covered today. Do all animals have complex tissues, brains, and blood?
The answer to that is a resounding no. It's so easy to assume all animals are built like us. But remember our very first case study. Sponges possess absolutely 0 true tissues. They rely entirely on just 2 layers of independent cells to pull off all their life functions. And on top of that, animals with radial symmetry, like those cnidarians we looked at, completely lack cephalization. Since they don't have an anterior end, they don't have a distinct head or a centralized brain. So being an animal definitely doesn't automatically mean you have complex organs.
Let's do one more quick formative check. Try to recall those blueprint definitions we established earlier on the drafting table. How would you compare the physical symmetry of a typical cnidarian, like a jellyfish, to that of a mollusk, like a squid or a snail?
Well, if you remembered our definitions, you'd know that cnidarians exhibit radial or biradial symmetry, meaning they're organized around a central axis. Mollusks, on the other hand, belong to that massive bilateral clade. They exhibit bilateral symmetry, meaning their left and right sides are perfect mirror images. This setup perfectly suits their directional movement and those true coelom body plans we talked about.
Section 6 Concept Recap.
Awesome job making it through. Let's quickly summarize our tour of Kingdom Animalia. First up, the basic bricks. All animals are multicellular, heterotrophic organisms, and their cells completely lack rigid walls. Second, when we classify these animals, we look straight at their fundamental blueprints, specifically their symmetry and whether or not they form that protective internal cavity called a coelom. We saw that sponges, porifera, represent the simplest architecture out there, asymmetrical animals completely lacking true tissues. Moving a step up in complexity, cnidarians utilize radial symmetry and have those 2 unique life stages, the polyp and the medusa. Finally, we checked out mollusk and annelids, which are these highly successful, bilaterally symmetrical protostomes that use a true coelom to achieve incredible biological flexibility.
As we wrap up this explainer, I really want to leave you with a thought provoked by this final question. We've seen how even simple changes in a biological blueprint, like say adding a colome or concentrating nerves into a head, can lead to just a massive explosion of successful new life forms. So, if evolution is the ultimate never-resting architect, what entirely new biological structures might be drafted in the millions of years to come? It's a pretty fascinating question to ponder. Thank you so much for hanging out with me on this tour of Kingdom Animalia. Keep asking questions and keep exploring those blueprints.